Welcome to another episode of the Dendrite Podcast with us, Saeed and Farno. This is a conversation with Dr. Emilio Alarcon. He's an associate professor at the University of Ottawa Faculty of Medicine, the director of the Biochemistry Graduate Program, and the Bio-nanomaterial Chemistry and Engineering Laboratory at the University of Ottawa Hart Institute.
His research focuses on developing bioactive materials and nanotechnology for tissue engineering and wound healing applications. He combines biomaterials and regenerative medicine expertise to explore innovative approaches that address practical and medical challenges.
Dr. Alarcon collaborates with researchers, clinicians, and industry professionals to ensure his work has real-world impact. His contributions to the field have earned him recognition within the scientific community, and he's actively involved in mentoring students and fostering interdisciplinary research.
We hope you enjoy this episode, and now here's Emilio Alarcon.
Hello, Dr. Alarcon. Good morning. So let me start by asking you this question. How do we save a failing organ with poor regenerative capabilities?
So the easiest way to save a failing organ is organ transplantation, right? So this is the end of the stage solution. Now, to save an organ that is not functioning well, you have to repair it and try to bring some of the functionality back. Mm-hmm. Now, most of the time in the space of tissue engineering, you are not really looking into save the organ, but provide some functionality back. And why this is important? Because, when you have patients with one organ that is failing, for example, the heart, and you like it or not, as the disease is progressing, you're going to have other organs fail. So when you have multi-organ failure, most of the time, those patients are not candidates for organ transplantation. So it's all about you can stabilize an organ so that patient will become eligible for an organ transplantation. So there are technologies that you can directly go and intervene, or you can actually reduce the workload on that particular organ. That is the reason why sometimes you have hemodialysis interventions like this. So it's about just making sure that the organ is functioning to a level that is suitable for the patient to have a good quality of life.
So at what stage do we start thinking about intervening and with tissue engineering at this point? Like, does it have to be a multiple organ failure, or which one is in priority, like a transplant, or if it's still like fine in terms of kidney with dialysis, do we still go with that? In terms of options?
It really depends on the quality of life of the patient, right? So, if even though the cornea is that an organ, but it's part of it's a tissue within the eye. That's a particular good example because you will intervene as soon as possible because, a damage in the cornea would let you blind or with partially sighted. And so even though the organ is still functioning, like as a whole, it's stable. What you have on the corner of the surface is limiting your ability to see and to perceive the world. And so in that particular case, you would like to intervene as soon as possible. But everything is tied to when can you intervene, and that is also dictated by the best practices in the field. Right? So certainly tissue engineering is made available to the physicians and the healthcare practitioners, but we are not the one actually deploying this. We are really working with them. So it's a multifactorial thing. It depends on what is available out there and what is the patient need, and also what are the best practices within the different realms of medicine you have available today.
Mm-hmm. Would you deep a little bit about what kind of method you use to assessing the efficiency of those things?
In terms of the regeneration?
Yeah. Like, how do you assess if it's successful or not?
It depends on the organ. For example, if you are treating a heart with a reduced ejection fraction, which is basically the amount of blood you're pumping every time your heart beats, right? and you're treating that, with a direct injection within the heart muscle. And you're expecting this to improve the amount of blood that is being pumped every time the heart beats. Of course, you can run an echo and you're going to see it right away. But it's a little bit more complicated. For example, if you are trying to regenerate tendon or like some muscle, you will need to wait a much longer amount of time, right? Because you have to stabilize the piece of, like the limb or whatever thing until the healing is completed. So, if you're regenerating the cornea, right? So, if you're transplanting a biosynthetic cornea, you will see it pretty much in a week or 2 weeks if the patient can really see through or not. So, but you have to separate what is technically the experimental way to the patient experience, right? Back in the days, I participated in this clinical trial in Ukraine. And when we treated patients with ulcers, very severe inflammation in the eye, we replaced the cornea, and it was a great success. But several of those patients, they were already blind because of the nerve damage. So they were still blind after the implantation. And they were happy because, the ulcer produced so much pain in them. And so in that case, it wasn't about the functionality of the organ, but more that pain relief. So they could actually go get a good night's sleep. They could go for walks and stuff like that, even though they were still blind. But the pain of the ulcer, it was gone. And so everything I hear around is the patient actually experience and patient well-being.
Yeah, it's just amazing how many diseases we can solve with this kind of treatment. I'm just wondering, so what is it actually happening currently in your lab and what you're working at the moment?
So as everything in life evolves, right? So, back in the days, we were working in recombinant human collagen, which is a type of collagen, that protein, a structural protein for pretty much every single mammal or living organism on this planet. And that was produced by, small organisms. So it was high quality. You could use it in humans. But it was really expensive. It was really difficult to maintain. So what we had to do, we moved to peptides, which are a small part. It's like small sections, like the Legos that form like a big structure. Like, imagine like the protein is composed by many of these Legos components in there. So we're using peptides for that. And the good thing of the peptides, they provide you a lot of functionality, the ability to manipulate what is in there, when it's in there, and how it's in there. So having those three things in there is providing you a lot of, like opportunities, right? So, we can say, okay, in the third position of this peptide, we want this functionality that is going to be specific for improving adhesivity, which is a critical property for, wound healing. Or you can say, I want in the third position of this amino acid something that is anti-inflammatory, which is key for cardiac repair. Or you want to have something that is basically providing you additional structural support, which is critical when you want to develop solid implants like corneas and stuff like that. So it's very interesting, and that is what we're doing now. So we're trying to find ways to produce better materials by using peptides. And also peptides are very relatively easy to scale up, right? And they're cost effective. You can develop peptides at a stable room temperature because it's not about, developing the therapeutics, but making sure that this therapeutic can be deployed, anywhere. You don't need to have a -80 freezer in your hospital that many places here in Canada and in remote areas in Canada, they don't have. If you develop something that is stable at room temperature, that is even better.
So more on that note, what are the main challenges of biomaterial development from a chemical and a biological perspective?
So for biomaterials, if you're working with naturally occurring polymers, or polymers, to be honest with you, is the batch-to-batch reproducibility, right? So let's say that, you are extracting this collagen from cows, from bones, or from skin. They're all different, right? So you're going to have a big variability. From a biological point of view, safety. Safety is always something that is challenging, particularly thinking about the regulatory normative and all the tests you have to pass. And you like it or not, when you're using animal-derived collagens or animal-derived proteins, it's always the risk of, cross-species contamination. It's very small, but it's still there. So it becomes very expensive because you need to have a certified facility, processing plant. So it can cost you several thousands of dollars. Just to give you a number, like a recombinant human protein, like a good quality collagen, it could cost you up to $15,000 a gram. So if you need 1 gram for treating one patient, it's automatically around $30,000 treatment per injection. But when you're using peptides, you can go to a fraction of that. So it is challenging, but there is a lot of, everything what I'm doing now in my lab is thanks to my collaborators, but my colleagues. They have done decades of research work on using naturally occurring polymers. So we're technically building on what actually they have been doing. So this is fantastic. But at the time of trying to move that to the clinic is a complete different story.
Right. Can I just ask, what would be the source of that funding? Where does it come from?
I mean, in Canada, we have universal healthcare, right? So whatever technology you put out there had to be uptaken by the healthcare practitioners, and it's an entire system, right? So Health Canada is very involved in this. It's not like you go and I'm not going to put this. It's a regulatory pathway. So just to give you an idea, for dental implants that classify normally as class 1, biomedical device, that is the name they have, this material, biomedical devices for the most part. It takes you anything between 5 to 8 years from the moment you develop to the lab to when it's in the patient. The more complex the organ or the tissue you want to treat is, the longer the life, the time of the regulatory pathway. So normally if you want to develop something for treating the heart, it can take you up to 25 years. This is still created until it's in the patient. So it's very lengthy because it's very expensive. Now, in Canada, we have the TRIC Council. So normally these clinical trials are funded through, the Engineering Council or the Medical Council. But it's very expensive. So, I mean, I'm going to give you a ballpark number. So you're looking at anything between 100 and 550, $700 billion just to mobilize one technology, right? Of course, we don't get that amount of money. It's impossible to get that amount of money. So normally we work on all the initial development and optimization. So we have to create a patent. We need to partner with private sector, and then we need to go to the clinical trials. So big companies, they normally come in after you have a phase 1, like a safety trial, and you show it that your material is safe. They will come in. The private companies, they normally, very rarely, they come before that. Yeah, it's expensive.
Yeah, okay.
And what would be a fundamental difference between using natural material or using other materials such as silicon, for example, like the implants of Neuralink, in terms of cost, in terms of viability, in terms of even use?
I'm not very familiar with Neuralink, to be honest with you. I lost track a couple of years ago. Mm-hmm. But what I can tell you is that silicon, they're not really active, the pieces you're implanting, which are completely fine if you're doing, cosmetic surgery, like surgical repair, the face and different things. Now in the space of tissue engineering, what you're looking into is promote endogenous repair, meaning that you want to actually re-instruct the body to repair itself, which is very complicated and very energy demanding. So the benefits are, when you can regenerate, for example, the cornea, right? And that is great because you will need to actually remove it. Well, when you implant something that you know that is not going to be fully integrated, eventually you will need to remove it. That is a little bit the caveat here. And let's say if I implant a device that is going to be monitoring your brain function, eventually you will need to replace it, right? So in tissue engineering, what we don't want is to do that, right? When you can. Of course, I mean, if you're performing a surgery in a very elderly patient where the stem cells, they're very lacking and all the endogenous repair is very slowed down. It's not existent, but it's very slowed down. It's going to be much more challenging to achieve that. So again, it's a compromise of what do you want to get and when do you want to get it.
Let me just ask you now you already mentioned a little bit earlier, but for our audience who are not that familiar with this topic, what does it mean for therapy to be translational? And if you can explain a little bit more on that.
So normally we like to say translational research, right? Or translational therapeutic, meaning that you develop this in the lab and eventually it's going to be used in the clinic. Now, that is easy to say, but very difficult to achieve. So last year or the year before, I can't remember, we screened the literature for materials to repair cardiac tissue, right? And I think we funded 20,000 or 7,000. And out of those thousands of papers, only 8 or 10 moved to clinical trials and 0 are in the market. That is not surprising, right? Because it's, most of the time when you are developing things in the clinic and you really have that clinicians working with you from the onset, it's very likely that you won't be able to translate that, right? Because it's, working in an operating room, working with patients, understanding the disease, seeing what is needed from the population is critical for developing technologies that can be translatable to the clinic. It's not the other way around. And there are very few centers around the world when you have in the same building clinicians and basic researchers, basic scientists. And there are even fewer buildings within those buildings that you have that direct, interaction. And that is the most difficult part to achieve because particularly in Canada, right, when the healthcare is universal. So all physicians and all healthcare workers are all the time taking care of our patients, right? So you and we are not like in the States. You have a big center where you just bring clinicians, you take them out of practice because they're more than enough, we like it or not. In Canada, every minute of our clinicians is very precious. So that is the one risk, but at the same time, the one advantage for the very few centers that they are able to do that, not interacting only, but collaborating and eventually doing this transdisciplinary science.
Right. The one topic that I've been really kind of interested in for the last couple of months, I guess, is the placebo effect. This might not be related, but I'm just wondering, like, for example, for other drugs, when we have the trial and want to see if the drug works or not, we use the placebo. Has there been any work with cell transplantation that we assess placebo effect, or if not, would that be a possible opportunity to see how does that affect?
I mean, it depends if you're doing an off-label, open label, case or something like that. Let's say that you want to regenerate the cornea, right? All this is assuming that the person deploying the technology or using the technology, the physician doesn't know what is being administered or not. It could be the physician, it could be the nurse, it could be the healthcare practitioner. But if you're going to implant a cornea in a patient with an ulcer, you're going to see which one is actually the implant or not, right? So, but in clinical trials, for example, of cardiac tissue, you normally have the control group, which is, is randomized. So you will have just inject buffer saline, or you can be injecting something that is supposed to improve the cardiac function. So, Unfortunately, most of the time, the trials are very small at the beginning. So there is always issues with, like power, like how solid the stats really are. But again, after phase one, when you have big money coming in most of the time, if the technology is very promising, you will see a change in the multi-site. So they are running this in different sites in different countries. So it's there when you need not only the money, but you need the expertise of these pharma companies and big enterprises, because they're enterprises and not companies at this point. And that is a value there, right? Because they do this for a living. Now, the placebo effect, you could see it. And that is the beauty of this. So now, as long as the patient feels okay, it's okay. Now, if you are blind and I am implanting you something that is not okay, it's not ethical, right? So in that case, the surgeon will need to know. Now, for cornea, it's a completely different thing, right? So it's, it's no way that you will see the implant because you had to put it in. But it's a good thing.
Yeah, it just amazed me how big effect it plays in everything, and sometimes we're not aware of it, but that's cool. So we'll look forward to that future research about placebo and this kind of treatment.
And you already mentioned a lot about this, but I'm just wondering, what are the commonalities between different therapies for cornea or for cardiac tissue, and what are the more personalized aspect of it for every patient? So every patient. So like a personalized regenerative therapy or more generic applications for each. How can we divide this?
So now there is a good space for, organ morphology, right? So like tissues, some people call it four-dimensional. They, like they change with time. I would say that, like reshaping organs, particularly organs where the shape is super important, like the eye, right? So the curvature of my cornea is different than yours. So if I get like a corneal transplant, it won't be the same. So I won't have exactly the same visual accuracy. And that is a little bit of what some researchers, including my team, were working on, trying to improve not only the shape, but also the thickness and the curvature and all the to really replicate your eye. So we can just put a new cornea there with exactly the same features. So in terms of personalized medicine, of course, I mean, what you could do is if you have patients with, with underlying inflammatory conditions, if you want to treat the heart, you should put something that on top of the formulation should be anti-inflammatory. If you have patients with reduced vascularization, like elderly patients with microvascularization problems or challenges like diabetic patients, you can add in the implant again for heart or for limb or whatever thing. And you can add a little bit of like a pro-angiogenic, some molecules that really recruiting these cells to grow the vessel. Now, really at the more refined, personalized medicine, as people tend to believe, I think we are far away from that, but it's there when creating instructive materials where you can really finely tune the chemistry and say, okay, I want in this position this, that. I want in this other position this, that. Or in the surface, I want this. Or within the actual structure, I want this. Because this patient had this profile. I think it is there when having materials that you can really finely tune the composition, the chemical composition is going to be critical, because using naturally occurring polymers that you know what they are, but you cannot really go and manipulate the residue, whatever in that protein, because it's almost impossible at this point. It is there when I think that the big bag of opportunities is coming with using peptides.
Let me just ask you now about, so you teach the science communication class, and you've been around trying to help students get more involved and be able to be more skilled in this field. What is the most, or let me ask you this. What is one thing that students lack the most in scientific communication that you think we need more work on?
I wouldn't say that they lack of. I would say that there is room for improvement and they can learn, right? So everyone can learn. I think that the newer generation, they communicate differently, right? So when you're teaching, you're also learning. And I think that you cannot forget that. I think if there is room for improvement, certainly is, I would say that, the newer generations, they have to deal with the older generation, the much older generations, right? And I think it's there when they need to wait. They need to learn how to communicate with this intergenerational, realm of humans in this planet, right? So, just to give you an example, I refrain myself of teaching, like how to do social media, because I tried for a couple of times and certainly my students were better versed than me. Now, what I do, I just give them a little bit of, like input, like, tips in here. But I need to spend more time in teaching how to effectively communicate in written format, right? Because, in drawings and all the different things are better than me. That's okay. So I think it's about use for the learners. I don't like to call them students because everyone is a learner. It is to understand that we live in a world with multiple generations, right? And when you want to reach out to the community, you want to make sure to reach to those individuals. Just to give you an example, the average, what is the average age of individuals living with cardiovascular diseases, right? So it can span anything from 30 all the way to 80 years, right? So certainly the younger population is much smaller represented in there. So if you are working at the university level and you want to reach out, to those patients, certainly the language style, it will have different, brackets, right? So between 30 and 45, between 45 and 60, and about 60, right? So in reality, they need to understand that there is no one fit them all, but they need to position themselves as, okay, I'm going to reach to this particular population, then I'm going to reach to a different group and so on. And within those, you also have different cultures, right? And within the cultures, you have different subsets of cultures, and you have also different religious things in there, ways of life, you like to call them. So that, and I think that is embedded in their training and their environment. It's just, showing them, like, you are a scientist, but if you want to communicate this, you have to be aware of the diaspora of individuals you have out there.
Yeah, that's just amazing, because be able to communicate that science is one thing, and also communicating in different languages, for example, or different cultures, you mentioned, is another thing that we need to really pay attention. How do you like teaching more or research, if you had to choose? I'm just wondering. Or which one is your favorite?
Teaching. I like teaching, but I think that when you work in the lab and you also teach, right?
Right.
I like both. I like both, but I've been teaching for over 20 plus years, right? I was a high school teacher before becoming this. So I like teaching, and I think that is certainly one of the most impactful things I will ever do in my life, teaching. Yeah.
Right. So what are some opportunities that students can get involved in your lab at the moment? Are you taking any honor students, or is it just TMM? Yeah.
We're taking students, honor students who normally have some summer placement, summers. I'm also directing a create program, which one of these NSERC, which is, engineering based and science based training programs. It's called in biotech.ca. So you can go and check in there. We also have opportunities for summer internships and stuff like that. So,
right.
If students want to learn, it's my duty to teach them. Right?
Mm-hmm. So usually they would send out the emails by this time of the year if they want to get involved, right? Okay.
Yeah.
Cool. Awesome.
Now, on a more personal level, how's your son?
He's doing great. I think he woke up. He's running around.
So could you share with us the memory of the day he was born, or the way that you felt that day?
I had one. When I was reading that last night, I was thinking about it. So Alonso was born on a very it wasn't a rainy, it was a very snowy and cold day, right? It was the middle of December. It was around, I think, on the 21st, on the 22nd. So we went to the hospital just for a regular echo, right? It was an echo, a routine echo. I could barely speak the language, by the way. And they found out that it wasn't growing, and one of the umbilical cords, the vein, wasn't really pumping blood. So they decided we need to extract the baby now. He was supposed to be born in early January or mid-January, and it was the middle, like late December. So we went to Montfort Hospital, very nice, but we didn't want to have really gynecology, so we have a midwife, and she was fantastic. She was the most caring human being I ever met in my life. So she was with us during the entire process. So we ended up just having a cesarean, a C-section, and Alonso was born. He was a tiny baby, and you couldn't tell if we had Down syndrome or anything, right? Because he looks like me, right? Like indigenous, like my people back in Chile. So he was like slanted eyes. He was crying. So, and yeah, he was born there, he was born with a hole in his heart. So it was the scariest part. And Chio the cardiac surgery unit was full, I remember. So they had him ready to be like lifted in a chopper and in a helicopter and to be sent to sick kids for a surgery. But fortunately, I think that thanks to the cardiac surgeon trained guys at the Heart Institute, they were working. You check on him very quickly via the phone. So we have to put the stethoscope and put the stethoscope, like the sound through the phone. And they say that's fine. Just discharge the baby. Just keep an eye on the weight. And that was the day he was born. So he was born with a lot of paraphernalia, but he was a happy little one. He was super cute.
That's great.
So the decision that you made about changing the focus of your career from a industrial chemist to tissue engineering, was that just a powerful moment and a powerful decision at one moment, or was it more of a gradual realization of a new path and a new meaning?
It's difficult to say. I think that I never like doing the same every single day, right? So that's the one thing that it was training my life when I was working as a high school teacher. So I really like it, but at the same time, I was repeating the same. So what I will say about the question is, it's the gift of time, right? The importance of living the present and living in here. We tend to believe that we are, and that's true, right? We're immortal, right? So we have an infinite amount of time on this planet, but we don't. And during the time you have in here, you have to make sure to help others, and not to do something meaningful for yourself, but for others. And because we are in the situation we are, because people just keep doing things for themselves, and that is the reason, right? So we lost the connection with the communities, and we are part of the community. We are within the community. So I decided to do something meaningful the day that my son was a child. And when we were told that Children's Hospital of Ontario, he was having his 5th chemotherapy round. He's a leukemia survivor. And we were told that he was going to die, that nothing else he could do. And everyone in the room was super sad. But I was not, because he was there. And I had the gift of time. So he wasn't gone. He was there with me. And no matter he will always be with me. When I understood that love is the one thing that transcends this plane, transcends this life. And love to me was time. So I got to know him better. I got to be with him. It was difficult, right? And that day I say, I have to do something. I have to give the gift of time back to other people. And maybe I might not be the one you doing that, but maybe my students will do that, right? So my students, they will become healers. They will become like medical people. and they will care for others. And maybe my invention someday will give that extra 5 minutes, because even 5 minutes, even 1 minute extra of in this plane, in this planet, in this life, it might make the difference between you saying goodbye to someone you love or not. And you cannot take that thing away. So, yeah, that was it. I decided, you know what? I want to do this.
That's the most beautiful reason to kind of change your career. And I mean, it's hard. It has its own challenges and hardship, but at the end, I'm sure your son would just be really grateful to have a father like you and the amazing things you have done for him. So maybe if you can talk about what was going through your mind when you decided to, okay, I'm an industrial chemist, but now because my son is suffering from this disease or this problem in a hole in his heart, what were you thinking in that moment, or what were some challenges to be able to just change your career? I mean, it was kind of related, but if you can maybe explain a little bit more.
Yeah, well, first of all, we persons with disability, we don't suffer from things, and I would like to make that clear. Right? We suffer because of this. We live in a society that is disabling us. We are not disabled. The society is the disabling society, right?
Right.
And my dad has been in a wheelchair for 45 years, so I never got to see him walking. But for most of my life, I didn't even know he was a wheelchair. He was a paraplegic patient. The legs were his wheelchair, right? So it wasn't surprising, right? So I was always around disabled individuals, if you want to call it like that. So the collection of friends that my dad had, it was very peculiar, right? With all types of disabilities, including intellectual disabilities. So to me, just having my kid, it was like, it's your time now, right? So it's your time just to look after him and just make sure that you reconnect with those individuals that were that integral part of your life. And it was difficult. It was the most difficult part is that my wife and I were here alone, right? So we thought we were alone. I want to make that clear, right? We thought we were alone, but we're never really alone. Like people in here from all different cultures and ways of life, they were with us across, along all the long way, even to this day, right? So we have friends from all different places in the world. And challenges, I think that the challenge is the stigma, right? So the stigma of living with a disability is not easy anywhere. And we live in a country where we tend to believe that inclusion is real, but it's not. I call it the illusion of inclusion. But you know what? I think that having my son with me has reshaped me as a person, but at the same time has allowed me to be more vocal on advocacy and of course in training the next generation. So the suffering from it is not your fault, by the way. It's the system that has teach you that we suffer. But we don't. If you look at my son, he's a happy guy. He's a happy guy. He actually doesn't understand the sense of time. He cannot tell you that tomorrow is going to be that day or yesterday was the other day. For him, every single day is a completely marvelous new thing. Mm-hmm. And imagine if we were still able to see the beauty of a sunset, the beauty of a sunrise. We have forgotten that. And that is what actually the Creator was meant to us, to never forget that we are mortals and every single day is a gift. And I'm telling you, he got that very clear.
So you mentioned your father and the gift of time and how that relates to love. I was wondering, what can you tell us about chess and how does it connect you to your father and to your son?
So when I was little, I was a very weird guy, right? So I wasn't gifted with the ways of language, right? So I had very, very hard time like writing in Spanish. I can barely write more. But it was a weirdo. I define myself, right? I like very things I didn't need to interact with many people. So I learned how to play chess probably when I was 2 or 3 years old. I love it. And my dad, we didn't have much money. We had a very humble family. And so I had this cardboard made chessboard. And my dad is an artisan. And so when he saw me like playing the first time, he just taught me a couple of things. So he crafted a chess set for me from scratch. And that was the most beautiful one I had. So we started playing. I mean, I've been, playing with him. I played competitively back in the school. I wasn't the best, but it was the safe place to me, the place that I always I could come back and see my dad. And it was very good memories, actually. Very good memories. And when Alonso was born, actually, before I knew that he had Down syndrome, I was already thinking that he would actually be beat me at the age of 3. You see, that was my mistake. I was always thinking about intellectuality as the most important thing in life, but I couldn't be wronger, to be honest with you, right? And so, yeah, I mean, when he was born, I didn't know if I was going to be able to play, but looking back, I think that it wasn't about that. It wasn't that I want him to love me. And he loved me for who I am, and I love him for who he is. And that is the most important thing when you're a parent. Right? You love your kid no matter what. And you don't look for the kid to love you because what you have done for him or for her. Your kid had to love you because you are the person, right? So, yeah. So we don't play chess like the way people play chess in here. So he got a couple of things in here and there. He plays with a dragon and we go around. And at the end of the day, life is about being happy. And he's a very happy guy.
Well, we'll look forward to maybe or I think Saeed plays a bit better than me, but we definitely look forward for a time to also play chess with you and see if he can beat you or not.
I'm not that good guy. Don't worry.
Yeah, okay. Good. So yeah, you mentioned that we definitely need to understand the different aspects of it. And the system obviously plays a really important role. But I'm just guessing, so obviously your work is great to be able to share that knowledge with everybody else. But how can, as students at U Ottawa, as an undergrad, for example, be able to understand the different aspects of inclusion, and how can we first become more knowledgeable, but second, be also able to share that knowledge with everybody else?
I mean, of course, I'm very outspoken, right? So, and I'm outspoken because I'm speaking from my privilege, right? So I'm a position of privilege. Of course, I can say like I have autism and I have different learning disabilities without the fear of losing my job or being looked, in a different way. That comes from someone who has a tenure position at University of Ottawa. Let's start by that. But, from that position of privilege, what I can do is just making sure individuals they find a safe space where they can come and be themselves. And this is what I do in my lab. I never ask people in my lab if they have a disability or not. Everyone is welcome and everyone is supported to the best of my capabilities, right? And for students used to be more active or like advocating in this space, we live in a society that historically has marginalized us, right? So in Canada, around 27% of the Canadian population, they do have a disability. And by 2030, I think we're going to ramp around about 30%. We're still underrepresented in the disability space, in the academia particularly, right? Less than 5% or 3% of faculties at University of Ottawa, they openly say they have a disability. So you see, you have an order of magnitude 10 times less. How can we do? How can I improve this? It's going to take time, but I think already students are much more outspoken, right? And disabilities include mental health issues, right? So I think what the students can do is finding the right place, right? Finding someone who can advocate for them. If they cannot advocate for them, just find someone they can advocate for. Right. So I have advocated for many actually learners at the University of Ottawa and colleagues And I'm hoping that it's not for them to pay me back, but to pay forward. So someday they find someone, just advocate for that person. And also for those who are non-disabled, is our responsibility, as in my responsibility as a faculty, just to educate them, just to you provide the training, provide like things like that, and that's what I have been doing in the science communication. And so having discussion about disabilities, about ableism, about like systemic segregation, that it really happened. It did happen quite a lot until very recently in Canada, right? So I'm expecting this conversation to become more and more important. Like probably 5-6 years ago, nobody in this country spoke about residential schools, and we are now very open about residential schools. I hope in 10 more years we're going to be chatting about the atrocities that were inflicted on persons with disabilities, were institutionalized in this country, and that is a process. It's part of the healing process and really moving on.
Yeah, I think the big challenge is definitely the time that we realize, okay, maybe we are not that good. Maybe there's room for improvement. I think that's the biggest challenge. I mean, at least for my experience as well, because once you realize that you have some problems that you need to work on, it's not that big of a deal and you can definitely get better. But the big thing is realizing that. So we'll definitely appreciate that. All right. Saeed, do you have any other questions?
Yeah, so you did talk about this a lot, but I just love listening to you talk more about this. What do you think is the as a meaning of life? What are we here? What are we doing?
I'm convinced that we came here to this planet not to grow and to become better, but to make sure that we help other people in the world. So, and that is the meaning of life to me. The meaning of life to me is just do what I was supposed to do when the Creator sent me here. I'm making sure that I help a person, any person, every single day. I have a very simple life, guys. So it's about helping others. It's about making sure that we're creating a better world for everyone. So yeah, so maybe it's too simple, but it's I remember that I heard this in an eulogy. It's very sad, but it's true. It's not about the love you receive, but it's about the love you give. And there are very few people when the time comes for them to meet the Creator, whatever Creator is, they can say they love and the word love. If at the end of your life you could say that, you were a very successful person. And that doesn't actually include the properties, the cars, the jets you have, or the amount of money you have in your banking account. It's about that. It's about when the time comes, you're going to close your eyes and say, you know what? I was loved and I loved. And maybe it wasn't perfect. Because love is not supposed to be perfect. Love is supposed to be real. And that is a big difference in there, right? People tend to believe that it has to be perfect. No, love is not perfect because we're humans and we're imperfect. So, yeah.
Well, beauty is indeed in simplicity, and what's more beautiful than that?
Exactly.
Dr. Alarcon, this was such a meaningful and fascinating conversation. We really appreciate you being our guest here. Thank you. Thank you for being a guest in our podcast.
Thank you, guys.
Thank you for listening to this conversation with Dr. Alarcon. In this episode, we discussed tissue engineering, challenges and complications, equity awareness, and his view on the meaning of life. If you're interested in his work, don't forget to check out the link in the description to his website.
This is a Dendrite podcast. To support it, please share this episode with your friends and anyone else interested. And now, let us leave you with a quote by Dr. Alarcon. We're not here to grow and to be better, but to help the others. Thank you again, and we hope to see you next time.